cavemen were very long-lived. Okay. I ran across this. This is maps into a paper Deborah Judge, a postdoc, and I published a number of years ago. And we asked the question, what is the evolved lifespan of humans if they were simply considered as another primate species with a given body and brain mass? Okay. We found this. It's simple. Ann Bronakowski's doing something like this, except with fertility. But anyway, body mass here, lifespan right in here on a log scale. But anyway, this is the primates. Here's Homo, and so that's basically the bottom line is that you have predicted lifespan. You start back here with chimpanzees. This is millions of years ago. The different, Australopithecus, Homo habilis, and so forth. You get out here, the predicted lifespan as the evolved trait of humans would be 72 to 91 years. A big spread, but the bottom line here is that the post-reproductive, that is menopause, is a phylogenetic legacy rather than a modern development. Okay? There's arguments in the grandmother hypothesis, all this, that in fact, well, it's an artifact of modern society and so forth. But this is a different way to get at it, but this is how we went after it, and that is that if we just view ourselves as another primate, which we are, it would be 72 to 91 years. Now, I ran across this in a anthropology book a number of years ago. They had this, it was in this book right in here. And I go, my goodness, this is really interesting. These are cave portraits. It's in La Marche Cave in France. And I go, this was like a cave, cave sketches of the inhabitants of a cave. This is 12,000 years ago. Anyway, when you look at these sketches, you have a young girl here, okay, probably, I'm just guessing, late teens. Here's maybe a middle-aged woman, 40 years old, somewhere there. Now look at this. You have older people beginning to emerge. This isn't some 40-year-old that's just, or 20-year-old that's just had a hard life. This is probably a 50-year-old. And you walk up to there, maybe 60-year-old, but here's right in here. This, again, is an old man. It's not somebody that's just, really just had a hard life and they just look old. There's all the characteristics of sarcopenia and these sorts of things in this old man. And so that here we are 12,000 years ago, this idea that everybody died young, tooth and nail, and all this is simply not true. And so there may be rare, but nonetheless there are some that really made it to the most advanced ages. Okay? Number 9, we have biomarker of health in fruit flies. There's live, this is medfly. And so, by the way, health demography and health span is becoming, it's a big part of research in the NIH, National Institute of Aging. And so that we wanted, the endpoint typically with biology anyway is death. But there's many other endpoints in, life course. And loss of mobility and loss of, you have to use reading glasses and so forth. So anyway, the idea that we could bring model system, in this case the medflies, to bear on questions in health was really important. So what we did is monitor the individual male medflies throughout their life. It's unbelievable. But when you go to behavioral literature, it's all about, spider webs weaving and birds feeding, parental care. Nobody looked at the entire, how does behavior change, both qualitatively and quantitatively throughout an entire life course. And so anyway, we did this. And so that now, this is not a dead fly. It's what we call supine. We call this supine behavior. There's a dead fly, okay? Now, they end up upside down, momentarily initially, and then later, but they can get up and run around, and then they fall down, or they end up on their backs again, and so forth. It's progressive. So when you look at these event history charts, if you remember the basketball players, each line is an individual. And so you stack these up, so you have a survivorship right in here. This is age here. Now, you color code according to no supine, no sign of that at all, versus intermediate and versus advanced and so forth. Now, you see this is basically a time to death rather than age-specific phenomenon. And so you have a few early onsets, like this, but generally it's about 2 weeks before they're going to die, you see that onset. And so you see here is the model. Some die healthy, just like any number of reasons somebody died, but it's very, very few. Most transition to the unhealthy state and then they die there. So we see that this transition is universal, it's progressive, it's predictive, and irreversible. And the whole idea here is that now you can, you can look at both active life expectancy and total life expectancy. And so this would be the onset right in here, active life for these flies. And then you have total life expectancy out here. Now in human demography, as many of probably the students and certainly the gerontologists here know, it's a matter of compression and morbidity. This would be the less healthy state right in here. So the idea as you increase life expectancy, is this staying fixed so that you're just increasing the proportion of the whole life course that's in the unhealthy state? Or if this stays fixed, can you push this closer and closer so that you have very little time in this unhealthy state? I call it the salmon model. You no longer have to get sick before you die. You can go right from healthy to dead. Oh, okay. It doesn't work that way. In theory, if you took it to the extreme, that's where. Okay. So there's that. Let's have a couple more here. Behavioral informatics, and that is we recorded this. I don't think you can understand aging without understanding behavior. Behavior, whether it's in humans, the vices, the exercise, the diet, all that's behavior. And but with flies, the same thing. I mean, behavior changes with ages. Nothing else, they slow down. But also aging, it changes with age, but behavior changes the aging rate as well. Okay, people that chain smoke get older faster and so forth, so that changes. Now, so we developed a technology to monitor flies, individual flies, throughout their life course in micro time, 5 times a second. We call this behavioral monitoring system. We published this in PLOS, where do we publish it? PLOS 1. And anyway, there's a system, just a schematic. There's a camera, 2 lights here, and you have flies in, 9 flies and individual flies in these cages. Okay, let's get through that. There's some sense of the data. It's just massive. It's informatics level data when you do the math. I mean, it's just really millions of points over a life course. And so anyway, now, what do you have with behavior? It's like, I got this from a demographer in the Netherlands, Frans Willekens. And life course is really a series of events. You're born, that's the first event. And then let's just, at a course level anyway, you go to school, different grades, events all along the way, you get a job, retire, get married, all that does. Anyway, now you have a sequence. What else is in a sequence? Well, you have genomics. And so now you have the nucleotide sequence like this. Now, once you view behavior as a series of events, that is, changes into different behaviors, you have this sequence. You can bring genomics-inspired software to bear on these data. So now, this is what we did. And so, for example, this is a dendrogram where this would be this is age right in here, and this would be in 10-day intervals.